When we ask you about protein-protein interactions, what do you think about?
Maybe you thought of Michaelis-Menten enzyme kinetics catalyzing reactions in the cell, or maybe SH2 and the induction of signal cascades, or possibly antigen-presenting MHCs in your immune system identifying foreign pathogens. You all undoubtedly thought of something different. That's because protein-protein interactions encapsulate a large area, facilitating an enormity of biological processes, including gene expression, cell growth, communication, apoptosis, among a whole bunch of others. Therefore, evolving how we investigate these interactions is vital in order to further understand these processes and the disease states associated.
We are TU Dresden,
And our project is called Space P. To improve how protein-protein interactions are investigated, we came up with a method that evolves binding partners efficiently. By using simple organisms and cheap, easy-to-use materials, we are able to develop protein interactions, making them stronger and more robust.
The process will take place in a flask called lagoon. In here, modified phages and bacteria meet. Phages, as we know, are dependent on entering the bacteria in order to facilitate their own proliferation by using the machinery of their newly found host. The phages we will use lack the gene necessary to produce the entry mechanism protein P3. It is in fact the modified bacteria that contain this vital gene P3, but it is locked away in its genome, regulated by a special upstream promoter. If the phages want to obtain this necessary gene, P3, for their own survival, they will have to develop a key that fits the lock on the special promoter. When the special promoter is unlocked, P3 will be expressed, and the next generation of phages will be able to infect their host. The key that will fit and unlock the upstream promoter is a moldable protein, and the lock is a target protein of interest. When the moldable protein does not adapt to the target protein, protein-protein binding is weak, and the phages will not be able to proliferate, since they cannot unlock the promoter and lack the vital P3 protein. Those that are lacking P3 will remain non-infectious and be washed away from the lagoon. However, if the moldable protein fits to its target, the phages will unlock the promoter, produce P3, and allow the next generation to proliferate, further evolving this protein to the target. The viable next generation of phages will be able to infect their host, proliferate, and inevitably remain in the lagoon.
Wiggle, wiggle, wiggle.
So, without any human intervention, and in the timescale less than a few days, the continued selection pressure will keep viable phages in the lagoon, evolving a stronger protein-protein interaction with each new generation.
Wiggle, wiggle, wiggle. Wiggle, wiggle, wiggle.
what do you think about it?
What do you think about? What do you think about?
TU. What do you... We are TU Dresden.
And our project is called
Space Speed.
Marvin!